Hey y'all. So in the last video, we discussed the general idea behind the TCA cycle, and we recognized that during the TCA cycle, we complete the oxidation of glucose or other carbon sources down to carbon dioxide, and the electrons that we generate are carried forth through these two molecules, NADH and FADH2. We discussed how these are really important electron carriers. And within the mitochondria, their role is to carry electrons either to complex 1 or complex 2 of this 4-protein machine that ultimately takes those electrons and reduces molecular oxygen down to water. Okay, so in this video, we're going to explore that process a little bit more. So the mitochondrial electron transport chain consists of 4 big, huge protein complexes named complex 1, complex 2, complex 3, and complex 4. And each of these have their own more specific names, but in this video, we're just going to refer to them by their numbers. Okay, so the role of complex 1 is to take electrons from NADH, and those electrons from NADH get passed on to an important redox cofactor called coenzyme Q, or ubiquinone. So ubiquinone accepts the electrons from NADH and gets reduced to ubiquinol. Okay. Likewise, the role of complex 2 is to take electrons from FADH2, and those electrons are given to coenzyme Q to make the reduced form of coenzyme Q, ubiquinol. So both complex 1 and complex 2 play the important role of accepting electrons from these central electron carriers we've discussed to reduce this central redox cofactor, coenzyme Q. Coenzyme Q then moves through the lipid bilayer down to complex 3, which ultimately is responsible for taking the electrons from reduced coenzyme Q and passing them to 2 equivalents of this protein cytochrome C, which is shown here in yellow. Cytochrome C is a soluble protein that can then move through the intermembrane space and interact with complex 4, which is responsible for taking those electrons and passing them on to molecular oxygen to produce water. So in this 4-enzyme process, what we've been able to do is to pass the electrons from either NADH or FADH2 all the way down to oxygen in a very controlled manner. And the way that this works is by passing electrons through very specific and finely tuned electron carriers. In complex 1, this happens first by NADH passing electrons to the first cofactor, FMN, which stands for flavin mononucleotide. Once the electrons have been passed to FMN, they then get passed through a series of iron sulfur clusters and ultimately get passed on to coenzyme Q. A very similar process happens in complex 2. This we discussed is where the electrons originate at FADH2 and they're passed through a series of iron sulfur clusters, ultimately to the coenzyme Q, which gets reduced. Complex 3 is substantially more complicated. We're going to try to distill the really complex process that happens in complex 3 down to a more simple idea. So first, the electrons actually travel through 2 different paths from coenzyme Q. In the first pathway, 1 electron originating from coenzyme Q gets passed through a specialized iron sulfur cluster called a Reski iron sulfur cluster, then through a heme group, ultimately onto the heme of cytochrome C. That's a 1 electron reduction process. The second electron from coenzyme Q gets passed through the second pathway, where it goes through 2 different heme groups and ultimately down to a different binding site for ubiquinone, okay, for coenzyme Q. This is called the Q cycle, and we're not going to go into a lot of detail about how it works in this video. But Suffice to say, the electrons in complex 3 go down 2 different paths, and ultimately, all the electrons from coenzyme Q get passed on to cytochrome C. It just has to happen 1 electron at a time. Okay? Once cytochrome C has been reduced, it will then travel over to complex 4, where it passes through first this really cool binuclear copper site, which is composed of 2 different copper metals, 2 different copper atoms, and then it's passed through a heme group, and then ultimately onto the active site of the enzyme, which is composed of another heme as well as another copper. And it's the space in between the iron in the heme group and this copper where oxygen binds and electrons are delivered 1 at a time to reduce oxygen down to water. Okay? So this is the electron transport pathway in a nutshell. The other really important thing that happens during the mitochondrial electron transport chain is that protons get pumped against an energy gradient. So opposing the energetic gradient, they get pumped from the matrix of the mitochondria into the inner membrane space. Okay? 4 protons get pumped through complex 1. No protons get pumped through complex 2. 4 protons get pumped in complex 3, and 2 protons get pumped through complex 4, giving us a total possible of 10 protons being pumped. So if the electrons originate from NADH, they pass through complex 1, which pumps 4 protons. They then pass through complex 3, which pumps another 4, and then through complex 4, which pumps another 2. So electrons originating from NADH pump a total of 10 protons. However, electrons originating from FADH2 start at complex 2. And complex 2 does not pump protons, so the electrons flow through complex 2 with no protons being pumped, then through complex 3, 4 protons being pumped, and then through complex 4, 2 protons pumped. So ultimately, we have 6 protons pumped from FADH2, but 10 protons pumped if NADH is what originally interacts with the mitochondrial electron transport chain. And that's an important thing to keep in mind when you consider the total potential energy that gets built up when we pump protons. And it's that energy that gets pumped up that ultimately drives the synthesis of ATP, which we'll look at in the next video.